Hello friends, welcome back to our channel. A urine sample can be tested to determine whether a woman is pregnant. The number of colored bands on the test strip determines whether the result is positive or negative. The urine of pregnant women contains a hormone called human chorionic gonadotropin or HCG. HCG is produced by the developing placenta and has important functions in pregnancy. However, some of these hormone is also excreted in the woman's urine. Women who are not pregnant do not produce HCG. A pregnancy test is based on the ability of protein called antibodies placed on commercially prepared test strip to bind to the HCG in the urine. The test strip contains three zones of functions here labeled R, T and C to indicate a reaction zone, test zone and control zone. The reaction zone contains a type of antibody that recognizes and binds to the HCG molecules and is therefore called as an anti-HCG antibody. The letter E signifies that these antibodies have been linked to the enzyme that can participate in color reactions on the test strip. Like the reaction zone, the test zone contains anti-HCG antibodies. In addition, these zones contain dyes molecules that will participate in the color reaction. The control zone also contains dye molecule for the color reaction. The antibodies in the control zone are called anti-mouse antibodies. These anti-mouse antibodies will recognize and bind to the antibodies currently in the reaction zone because the reaction zone antibodies were made in a mouse. The urine of pregnant women yields a positive test result. To determine this result, the test strip is dipped in the urine sample. The sample which contains HCG is drawn up the strip by capillary action and arrives the reaction zone. The enzyme-linked antibodies recognize parts of the HCG molecule and bind to these molecules from the urine. The complexes of HCG and antibodies as well as the unbound antibodies dissolve in the fluid and detach from the capillary membrane. They are carried along the test strip by the capillary flow. At the test zone, other anti-HCG antibodies bind to the HCG portion of the moving complexes. The antibodies in these zones are permanently affixed to the strip. So the complexes that binds here now stay. The free antibodies continue to flow along the strip. The antibodies that escaped binding in the test zone have reached the control zone. Here, anti-mouse antibodies that are affixed to the strip bind to these mouse antibodies. In the test zone, the enzyme-linked antibodies catalyze a reaction with the dye molecule, which acts as a substrate and trigger a color reaction. The same type of reaction occurs in the control zone. In a positive test, 2 of 3 bands are colored, but in a negative test, only 1 is colored. This difference results from a lack of HCG and the woman's urine. In urine lacking HCG, when the sample flows to the reaction zone, the antibodies have nothing to bind to. The antibodies detach from the reaction zone and migrate alone up the test strip. The enzyme-linked antibodies arrived at the test zone. Because the migrating antibodies are not carrying HCG molecule, the immobilized anti-HCG antibodies in the test zones are not able to snare them. The antibodies continue to travel along the strip. The control zone contains anti-mouse antibodies that binds to the region of the traveling antibodies and thereby immobilize them. The control zone is an important region that helps distinguish whether the strips are working correctly. If the strip is working correctly and if the woman is not pregnant, the control zone will produce a colored reaction, but the test zone will not. Hope you liked this video. Thanks for watching.